Hey everybody, Mr. Murphy here. I'm going to run you through an abbreviated look at Chapter 8, which is our Introduction to Metabolism and specifically our examination of enzymes. So Chapter 8 starts with a examination of sort of broad topics of energy, and I think it's worth just very quickly talking about some of those before moving into specifically how enzymes play a role. But when we consider energy inside of living things, it's important to go big and to think about the laws of thermodynamics, so bringing in some physics to our already chemistry and biology-filled topics. So when you look at the laws of thermodynamics, basically what it boils down to is that energy can't be created or destroyed, only transferred between forms, and things have a constant trend towards entropy or sort of disorder in the types of energy that exist. And so meaning that as energy gets used and transferred, more and more of it gets lost in unusable forms. And what that creates is a situation where it kind of seems like life is highly unlikely. Life is all about organization and building up systems that work in cooperation and in very complex ways, which doesn't really fit the narrative of everything's trending towards chaos with regards to energy. But the truth is that within any closed system, this is possible through the use of energy to create these systems which provide that sort of stability. And so biological processes and just biology and life in general is all about using resources to maintain that order in order to sustain life. And while life is a somewhat temporary thing, as long as that energy flow is there and constant, organisms can continue to take advantage of it to create these layers of complexity. This comes up with the idea of free energy, and there's an equation or a variable here called Gibbs free energy, which looks at the basically available energy within a system. And so we look at the change in free energy as a way to understand what has happened within a system because of chemical interactions. And if a reaction has a negative delta G, a negative change in Gibbs free energy, then what that means is that we've lost free energy and that we're trending towards a more stable state. And so you can think of this as a measurement of stability. The more stable things are or become is because they have lost more of that available energy, more of that energy that was there that could have been doing work, that could have been altering things. As that energy is lost and things become more stable, that's associated with a lower G value. So some examples here of like diffusion, things diffusing outwards, a person diving off of a diving board, a chemical reaction that takes a larger molecule and breaks it into smaller, simpler molecules. Those are all examples of negative delta G interactions. And I think the person on a diving board is a pretty good example of this, and just thinking about like that person could have laid down on that diving board and retained that higher energy state. But you could look at that and sort of easily see with your own eyes how that's kind of an unstable system. So we're just waiting for the moment when that person accidentally falls off or rolls off or whatever. That's energy just waiting to be released when it's locked up with that potential gravitational energy. So you can think of all of these really as potential energy because of the more built up complex state of these things. There is potential for energy to come out of them. In 8.3, it introduces sort of the importance of ATP as a molecule that enacts much of this changing within the cell. We're not going to get into it much now because we'll circle back to this more when we talk about the cell and then again when we talk about photosynthesis and respiration, but I didn't want to just completely skip over it. So keep in mind that the cell is doing all kinds of different chemical work. There's transport work, so creating concentration gradients. There's mechanical work of physically moving things in order to enact change within the body. So there's lots of different types of work, and ATP kind of is in the center of all of it, providing that currency for energy exchange that's happening. In 8.4, this is where we actually talk about enzymes. So enzymes fit into this puzzle because what enzymes do is they enable chemical reactions to occur that might not ordinarily occur at that same rate. And so they vastly increase the rate of chemical reactions and make them much more easily to happen. So some terminology. Obviously, catalyst is anything that speeds up chemical reactions, and an enzyme is a protein catalyst. So when we look at enzymes, we want to think about that as something that's made out of a protein that speeds up chemical reactions. So here's an example of sucrase. And what happens here is sucrose is a molecule, it's a disaccharide. It always has potential to break apart into glucose and fructose, but generally at normal conditions, it's not going to. Right? If you sufficiently heated it or added the right pressure or changed the environmental factors in a number of ways, you could probably encourage it to happen more easily, but that's not really practical for living things. And so by utilizing an enzyme called sucrase, living organisms are able to put sucrose into a physical arrangement that makes it more likely to split into glucose and fructose, which might be helpful, for instance, in the case of digestion. So this is where activation energy comes in. Activation energy is the energy necessary for a chemical reaction to even want to occur. You have to remember that when the molecule of sucrose is sitting here, all of its atoms are quote-unquote happy with regards to how many electrons they have. However, the arrangement, the sort of large-scale arrangement of carbons, hydrogens, and oxygens is not at as stable and simple a state as it could be. For instance, if it was broken down into glucose and fructose, or if it was broken down even further into carbon dioxide and water, it could be even more stability and lower energy state. So why don't things just immediately go to their lowest energy state? That's this idea of activation energy. You have to put some stress onto these bonds in order to encourage them to break to begin with, because even though they could be at a lower energy state, in order to get there, the atoms would have to be sort of forced into a very unstable state, momentarily speaking. And so that's what activation energy is all about. And so here's a diagram you've probably seen before where we have some reactants and we have to input energy. This EA is the activation energy. And that puts the bonds into this transition state where now there's so much sort of chaos because of thermal or kinetic energy between the molecules and the atoms that it's much more likely for them to want to break one bond in favor of another because they're already stretched into such a chaotic form. And so the activation energy for a given reaction is how much do you have to push on these molecules from a kinetic and thermal standpoint. How much do you have to push on these in order to convince them to even want to break those bonds to form new ones? And here you can see the reactants A, B, C, and D have turned into the products A, C, B, and D, which are at a lower energy state. And so that's a negative change in free energy, which means that that energy has been released and that the products are now more stable than the reactants were initially. So that activation energy is a barrier. It's the reason why things don't just spontaneously combust and just, you have a candle sitting on your desk and it just explodes into all the heat and light that it could have potentially ever been. That doesn't happen because activation energy is maintaining these things in a somewhat stable fashion or a stable enough fashion, and it's not until we introduce something to destabilize them that a chemical reaction can even occur. So enzymes do this or facilitate this by lowering the amount of activation energy needed. And that's this process of catalysis. They have created a complex with a substrate, moving that substrate, manipulating that substrate, and encouraging the chemical reaction to occur. And here you can see a chemical reaction with the addition of an enzyme where the same reaction, the same reactants, same products, but we don't need nearly as much activation energy. And so enzymes are capable of accomplishing this such that the activation energy needed to make the chemical reaction happen is within the bounds of the energy that's available based off of the body temperature of an organism plus any nearby, ATP or other molecules that might be playing a role in the chemical reaction. There's a little video that talks about this that's pretty helpful. So the terminology of enzymes, we know substrate is the thing that the enzyme is grabbing onto. That's the reactant or reactants of this. When it binds it, we call that the enzyme substrate complex. That's important because we'll see in a second how the enzyme actually alters itself in a way during that process. Enzyme names typically end in ACE, so you can recognize those. And the active site is the location where it's actually doing sort of the grabbing and the fitting onto the substrate. So when an enzyme binds to a substrate, it's not doing so, it's not truly binding in a sense. There's no covalent bonds or anything. This is typically hydrogen bond mediated. And what happens is, as the substrate comes in, because of the chemical profile of this substrate, it's going to be altering the amino acids making up this enzyme. And you can see how this enzyme kind of hugs onto the substrate. The substrate actually causes the enzyme to change its shape. And here's a little video showing that as the substrate comes in, the enzyme folds on itself, being pulled on, manipulated by the chemical interactions with the substrate itself. So, as I said, that's typically weak bonds like hydrogen bonds. That happens very quickly. The actual conversion of the substrate to the product is a nearly instantaneous thing. And then the product becomes released. Now, why does the product get released? If the substrate comes in and changes the shape of the enzyme, that might be forcing the enzyme to actually like put some pressure onto the substrate. And that may be actually the action that lowers the activation energy. But as soon as that substrate breaks into its products, if this is something that's being broken down, maybe through hydrolysis, as soon as it breaks into its products, those products, with the broken bonds and shifted movement, now have a different chemical profile than the substrate did. And so the product is not going to have the same affinity for the active site that the substrate did. And so the product ends up being released very, very quickly, which then returns the enzyme to its initial shape, ready to grab onto the next substrate. So because enzymes emerge unaffected and back in their original form, a small amount can have actually a pretty huge metabolic impact within a cell. So here it is. We have substrates coming in, the orange and green substrates. They bind to the active site on the enzyme, forming the enzyme substrate complex. They're held there by weak interactions. This lowers the activation energy for the chemical reaction between this green and blue thing. And so now that reaction can occur, turning the green and orange thing into a blue and yellow thing. That blue and yellow thing now, that's a new product, has a different shape. It's no longer going to be fitting as perfectly with the active site, and so those products are released. That enzyme now is back into its ready-to-go state, ready to grab onto the next set of substrates. And you can see how when it did grab onto those substrates, they caused a change in the shape of the enzyme, which was probably part of the reason why it applied pressure to these things to lower the activation energy to begin with. So you can see how you kind of have to go like multiple times through this before the whole story of it kind of becomes clear. There's lots of ways in which enzymes do this. So they can orient the substrates to each other. They can actually stretch the substrate to encourage it to break apart. Excuse me. The active site may provide a microenvironment that favors the reaction. And then a rare case that we're not going to get into examples of, the amino acids actually within the active site may meaningfully participate in the reaction. But that kind of goes against what we generally say about enzymes. So enzymes can be affected in a number of ways. Having more substrate gives the enzymes more to work on. But at a certain point, the enzymes are going to be working as fast as they can, so you'll reach a limitation. We saw this with the activity we did in the back of the room. If the enzyme becomes saturated, that's when we say the reaction rate is maximized. So saturated enzymes means that they are working as fast as they can, that even if you had more substrate, it doesn't matter. The enzymes are working as quickly as possible. Lots of other factors can influence enzyme activity. pH and temperature, for the same reasons as every other protein, can manipulate the shape of these things, which could have negative or positive impacts on how effective they are. For this reason, we would expect to find certain enzymes that are, optimized for certain temperatures or for certain pHs. So here's optimal temperature for a typical human enzyme is here right around 30 whatever Celsius, 37 Celsius, which is about body temperature. Meanwhile, if you are a thermophilic heat-loving bacteria in the nearby volcano system, you're going to have a much higher temperature, and therefore the same enzyme or similar enzyme within that organism may be optimized for a much more extreme temperature. which is to say the exact shape of it is going to be accommodating for the fact that at higher temperature you've got a lot more movement and flexibility to the molecules that are making up your enzyme protein, and so you need to accommodate for what's the standard amount of energy that's present, and how can we maintain more or less the correct shape in the face of that kind of external environment. Same thing with enzymes and pH. Typical enzyme in your intestine is going to have a optimal pH somewhere in the 7 to 8 range. Meanwhile, pepsin in the stomach has an optimal pH of 2. Why? Because it's in the stomach, right? And that's where we have hydrochloric acid. So again, some pretty straightforward examples demonstrate how enzymes can really be evolved to a particular environment, both within one organism or between different organisms. We have cofactors and coenzymes. Cofactors and coenzymes, this is all the stuff that you find on the side of the box of Cheerios, and you wonder, like, why is there zinc in my Cheerios? Well, turns out that cofactors and coenzymes help our enzymes to bind to their substrates. So these are, they may be permanently binding to it. It may be reversible with the substrate, but in some way it is facilitating the action of the enzyme. Enzyme inhibitors, meanwhile, can do the same thing but in reverse, that they bind to the enzyme and now they prevent the enzyme from binding to its substrate. And there's two types of that. The first is competitive, and here we see an enzyme and a competitive inhibitor competes for space at the active site and blocks the substrate. Meanwhile, we could have non-competitive inhibitors, and so here's that same enzyme, and this time it's not blocked here. There's nothing competing for the active site, but a non-competitive inhibitor has bound to the enzyme somewhere else and bent it out of shape, causing the shape of the overall protein to change, and now the active site no longer fits the substrate properly. And so you get reduced or completely shut down functionality of that enzyme. There's different toxins that are actually irreversible enzyme inhibitors, and a pretty dark example is sarin gas, which has been used in a variety of places horrifically. But what it does is it binds to the active site of acetylcholine esterase, which is an enzyme. Acetylcholine esterase recaptures the acetylcholine that's released from our nerve cells. So that way when you send a signal, let's say to your muscle to flex or whatever, the acetylcholine goes out and sends that signal to the muscle cell. And then the acetylcholine needs to be recaptured, so that way we can send another signal or stop sending signals or things like that. So acetylcholine esterase is responsible for that. But when it's inhibited, now suddenly we have no more control over acetylcholine that gets released between our nerve cells and muscle cells. And so that can be very quickly extremely devastating to wide-scale body functions. So pesticides and antibiotics include very similar things. Wasp sprays, I believe, function most typically in this way, which is why, if you blast a wasp with some nasty spray, the whole thing just drops out of the air because very quickly its enzymes are just significantly inhibited, which is like a bummer for the wasp, but not a bummer for you not getting stung by a wasp, I guess. The enzyme evolution is a fascinating one, and one that hopefully comes up in the back of your head, because as you're learning about enzymes, it all sounds so convenient that, like, they have just the right shape to grab onto just the right thing, and not only that, but when they grab onto it, it changes the shape of the enzyme in just the right way so the enzyme can change the thing it's grabbing onto. Like, it's so perfect. Like, how does that work? And it turns out that the same way as literally anything else within life, that the slow change and selection process over hundreds of millions or billions of years have led to some pretty complex and cool molecules being possible. And there's an example demonstrating this using E. coli, that a particular enzyme in E. coli resulted in a changed affinity over a period of generations for a particular sugar molecule. And so when they examined this, what they found was that after repeated generations of this thing, when they finally saw that it was producing this enzyme that was more effective, they found a whole bunch of amino acids, particular amino acids that had been flipped and tweaked and changed in such a way as to now produce an enzyme that's more favorable. And you can imagine how just by chance, a couple little mutations within a bacteria could result in a new individual that now produces this enzyme better, and how if it's producing it better or producing a better version of it, suddenly that's the bacteria that's probably surviving better, which means the next generation is going to be made up of more of that bacteria's descendants. So anyway, that's the whole story of evolution in a nutshell, but we'll get into all of that with a much later unit. Anyway, the evolution process of enzymes is certainly a fascinating and pretty cool topic to look into. The last section, just to talk about briefly, is the control mechanism for this. So we can have allosteric regulation, which is when we have a molecule that binds to an enzyme and activates it. So here's an example that an activator comes in and binds to this enzyme, and once that's present, it changes the shape of the enzyme in such a way that the enzyme can now grab onto its substrates. Okay. Now we could also have an inhibitor that does the exact opposite, that maybe the enzyme's constantly functional, except when an inhibitor comes along, it suddenly shuts down. So inhibitors and activators can work in tandem with one another and would work in kind of an obvious way based upon their name. Cooperativity is an interesting example where you have one substrate that comes in and that causes a change in shape of the enzyme that allows the rest of the substrates that it might be acting on to bind. Not going to be tested by us, but it's a cool topic. And then different types of feedback come into play. We will separately go through feedback mechanisms as we get farther into cellular processes, so we don't need to worry about this too much, but the long and the short of it is that enzymes are heavily involved in different feedback loops where a product is made and the creation of that product ultimately leads to a shutting down of the process that made that product. So the idea being that if your body needs X chemical, it goes through a whole bunch of steps and produces X chemical, and the production of that chemical goes back and inhibits the creation of it to begin with, because once you start getting the thing that you have, you want to send a signal that says, okay, hit the brakes, we've got it, we made what we need to make. So enzymes are heavily involved in that, but again, that's a topic for another day. This also feeds into future units when we talk about different membranes within the cell. Compartmentalization of the cell helps a lot with sequestering certain enzymes in certain places. So you can have something like the lysosome that breaks down like everything inside of the cell because it's full of these crazy digestive enzymes, but that's contained, and therefore we have some control over how and when that is exposed to things that we might want to break down, but not be exposed to things we don't want to break down. In eukaryotic cells, we see this a lot with different organelles that are housing specific enzymes. Lysosome's an example, but then also like in the mitochondria or the chloroplast as well. It's an entirely different ecosystem for them. So, kind of putting all of this in one thing, we have enzymes which alter the amount of energy that's needed. At the end of the day, what is happening is we're getting some reactants that are transitioning to a lower energy state. Most often they could be going to a higher energy state. That's a different kind of reaction. We'll talk about that more later on. And then the book ends with this picture of penguins, which I thought was nice, so I left it in. All right, if you have questions, please let me know.